Hello, ladies and gentlemen, and thank you for joining our webinar by OI Analytical, Hank Hahn. Hank is our Senior Sales Specialist. He's based in our Texas office, where it's currently 8 p.m. on a Thursday night. Hank, thank you very much for joining us today.
Thank you, Michael.
And I would just like to share with the audience that as of tomorrow, Hank will be celebrating his 29th year with OI Analytical. Congratulations, Hank, that's fantastic. And before I let Hank begin here, just a reminder, and for those who are first-time GoToWebinar users, there will be a question and answer period that will open up at the end of the webinar. However, you are welcome to type in your questions throughout the webinar in the chat question box on your GoToWebinar interface. We will follow through those one by one at the end of the webinar. Any questions that we don't have time to answer will be followed up offline by Hank. He will have a copy of all the questions as well as the email address of the attendee who asked it. And finally, a link to the recording and the presentation, as well as a certificate indicating your participation in this webinar, will be sent to you in a follow-up email after the webinar. And without further ado, Hank, please take us away.
Thanks again, Michael, and welcome everyone. This is our part 2 of the series on the model FS3700 Automated Chemistry Analyzer and Chemistries. So today what we'll be covering is going over the FS3700 Auto Analyzer, talking about the FS3700 Chemistries, and paying a little attention to cyanides and ammonia TKM by gas diffusion. Those are some of the most popular chemistries that we sell, so wanted to cover that a little further. I'm your presenter, Hank Hahn. I'm currently the OI Analytical Senior Sales Specialist. I'm based here in College Station, Texas. Nearly 29 years of analytical instrumentation experience, including all our product lines that cover gas chromatography, the automated flow chemistry, TOC analyzers. Sales colleagues refer to me as a valued resource, provide wealth of knowledge, not only my understanding of the technical side of our instrumentation, but also understanding how it best provides solutions for various applications in numerous industries. So what we'll start with is the FS3700. We like to work with customers' problem statements in developing instrumentation and then also in bringing something to market. So wet chemical methods for ion analysis are inherently very sensitive and offer analysts the analytical capability that is demanded by regulatory methods. However, these methods are also inherently tedious, time-consuming, and generally require large volumes of chemical reagents and hazardous waste. Problem examples for ion analysis with contract service laboratories. They've got a wide range of sample matrices and required chemistries. With your municipal drinking and wastewater laboratories, there's groundwater, surface water, and treated water with typical ion concentrations in the parts per billion range. We also have the chemical and petrochemical laboratories with treated processed water contamination levels in the parts per million. And they need rapid cyanide or other chemistries with on-site lab where they can get their numbers fast. As I mentioned last week as an example, there was a firm here in the Gulf Coast, and they were sending their samples to another facility that could take several hours and such for them to get results back. And by having this instrument on site, they were then able to get results immediately, see if they treated it properly, or if they had to retreat their wastewater, if it was at the right amounts and such, then they could release that. And so that's what's nice about having one of these instruments on site. So the model FS3700 platform offers the chemist the flexibility and analytical capability to meet their needs. It's truly plug and play. Has a unique design features that offer high sample throughput and low cost of ownership. We have robust technologies to meet the demands of every type of sample matrix. It's got excellent precision and accuracy, and we follow regulatory compliance to ASTM, ISO, standard methods, and also the US EPA. So with automating wet chemistry, The FS3700 is an advanced continuous flow analyzer designed to improve laboratory productivity by automating wet chemistry test procedures. What we've incorporated into the instrument to help with lab productivity is a FIA valve. Automated injection valve is installed in the analysis module chassis when required to run a flow injection analysis in flow injection mode. And a lot of our chemistries are in flow injection, or we also have them in segmented flow and can develop as per as the client has expectations. Each chemistry cartridge is pre-assembled with all the components needed to perform a validated analysis method. You just attach the pump tubing and detector flow cell. Ordering by the channels that we provide gives a convenient way to configure this 3700. It's modular, flexible hardware, provides a great platform for research, in-house or proprietary methods as well. We have the inline devices for reactions requiring heating or UV digestion, and they're mounted on the underside of the chemistry cartridges. Our FlowView software provides user programmable control of the UV lamp and also the cartridge heater set points in 1°C increments. We have the Photometric and Amperometric detector modules plug into the FS3700 to support methods employing colorimetric chemistries or amperometric measurements. And with the Amprometric detector, those are cyanide specific. Our expanded range photometric detector and auto scaling software virtually eliminate all off-scale samples, and a single calibration curve can range from low ppb to high ppm concentrations. We come up with value propositions as well. So the Flow Solution 3700 automated flow chemistry analyzer introduces state-of-the-art technology with a flexible, innovative system that brings together advanced icon-driven software, and it's modular, plug-and-play components that allow you to easily customize your system for your chemistries and improve your laboratory workflow. Designed for ease of operation and low cost of ownership, the system provides high sample throughput while handling complex matrices and a wide variety of chemistries. We also have one for our cyanides. We're recognized as one of the leaders in cyanide analysis. OI Analytical designs accurate, high-performance benchtop and online instrumentation for performing flow injection cyanide analysis on drinking water samples and wastewater samples from mining, metal plating, and other industrial operations. Again, it's designed for ease of operation, low cost of ownership. The system provides high sample throughput while handling complex matrices for cyanide analysis. So we'll talk a little bit about the FS3700 layout. With our features, for years laboratories have turned to OI for accurate, reliable, continuous flow analyzers, and the 3700 is the next generation. We built incorporating the strengths of our previous platforms from the Alchem product line that we purchased, our CN Solution, FS3100, and Flow4 product lines. It has the ability to run segmented or FIA methods. We've got a new user-friendly interface using the best of prior software. The previous version, WinFlow, now we use FlowView. Compatible with Windows 10. It connects via USB and can be run from any computer and some laptops and tablets as well. Chemistries available, which we'll detail a little bit more in upcoming slides. We've got available cyanide by OA 1677. We have available cyanide with ASTM D6888. That's the chemistry that can help with sulfide abatement. We've got total cyanide analysis with ASTM D7511. We have ammonia TKM by gas diffusion. We also have ammonia phenate nitrogen. We've got nitrate nitrite, your phosphate in all forms. We also have a low-level phosphate cartridge that gets down into the lower ppb levels. We do both phenol post-distillation and phenol with inline distillation, chloride, sulfide, cyanide with ISO methods, and also via post-distillation in case the regulatory agencies and such might not allow the distillation-free apparatus that we have with gas diffusion. The chemistries released for the FS3700 are the best method. selected from all configurations developed for previous generation analyzers and user submitted methods. The best method will be selected for use on the FS3700, regardless if it is FIA or segmented flow, and chemistries may be customized as needed. Again, it's a modular, very flexible platform. We offer a 180 position autosampler and also a 360 position autosampler if you have a lot of sample throughput. Some features of the 3180 autosampler is you can have two 90 position trays or two 60 position trays with that as well. We have 10 position standard rack, 50 millimeter conical centrifuge vials with caps, a wrench station. The wrench station is located at the left end of the standards position in the back of the sampler base. Onboard peristaltic pump, 2-channel peristaltic pump moves the rinse solution from rinse source through the flow rinse station. Sample probe kit. The kit includes sample probe. Probe fits onto the C drive of the autosampler. We have a 24-channel pump, and that allows to hook up multiple chemistries that you can have where you can do the easy swap out and plug and play. So for auto analyzers, the Ismatec pumps use multicolored pump tubes that vary in the amount of volume that gets produced. And to allow for a continual flow of solutions, the pump tube roller heads gently rotate around the tubing material. The compression of the roller cams on the peristaltic tubing acts like a check valve in the pump, keeping liquid flowing smoothly in one direction and offering a repeatable and consistent flow with each revolution. Pump tubes are held in place, easy to remove Platen that allows for microcontrol of each pump tube, and these controls of the pump can also be controlled by the FlowView software. So here's an example of the pump Platen that we use. So we have the Platen itself, Platen extender. The tension lever in the upper right-hand corner can add the tension to the pump tubes. Pump tube fits between the extender and the pump tube collar. The Platen extender and tension lever and tube material is phthalate-free PVC or Tygon. And then we have a different variety of pump tubes, and these are used throughout each chemistry. They're kind of given the color like orange blue, orange green, orange yellow, red red, white white, and I'll show some examples of what we do in using those with these chemistries. There's the internal diameter and the flow rate at 40% mils a minute. So here's an example of ammonia TKN. We would use an orange-green pump tube for air injection, black for a depubler pull, a yellow pump tube for sample line pump, gray, and orange-yellow. And so we also have pump tube kits that are specific to these pump tubes, put in a little holder and such, and we sell those as TK, or you can buy 12 packs of pump tubes. So what's in a channel? Everything needed to perform analysis is included in a channel. We include our FIA valve, chemistry plate, detector, amperometric or photometric, dependent upon the chemistry. The cartridge includes your gas diffusion modules, heaters, or UV lamps as needed. We include all pump tubing and other tubing coils with the chemistry kit. So as a breakout here of a colorimetric chemistry with a photometric detector, we have our FIA valve, and again, that's chemistry specific. We have our chemistry plate with the mixing coils. We do have, in this example, a debubbler, and then the photometric detector, which includes the filter specific to the chemistry. So if it's a 550 nanometer filter, our range was 420 nanometers to 880 nanometers. And what's in a channel with the Amprometric cyanide chemistry? All of our cyanide chemistry use flow injection, so you'd have your FIA valve, your chemistry plate with the mixing tees and pump tubes, the gas diffusion module, and then our amperometric flow cell with reference electrode. Our, excuse me, our amprometric detector, the cyanide chemistries only, excuse me, is the best detector on the market and has been further refined to reduce noise. It's not a third-party detector. We actually manufacture that ourselves. Excuse me. Improved cell design and upgraded electronics. It's made up of a reference electrode red, working electrode white, and a counter electrode black, which closes the loop. Off to the right, we have the gas reference electrode, and then also this is the flow cell for the amperometric detector. That can be maintained, and the electrodes can be polished if you start to see kind of a degradation in the response. With our photometric detector, it's the expanded range detector. It has a 10x increase in signal to noise for some photometric methods, improved equivalent detection limits from our prior model, the FS3700. It has lower MDLs down to low ppb, and limitation is the flow noise. With our detector, it does eliminate the need for auto dilution. It has a wide range capability. The benefit of using a wide range detector lies in its inherently high sensitivity. Because of its high sensitivity, it can detect very small changes in the signal without increasing the gain. And this allows the detection of very low concentrations. Because the gain is not increased, it can also detect high-level peaks extremely accurately in the same run without topping off. So we have a little breakout here of the photometric detector. There's the flow cell, 5mm is standard, and then we have a photo diode lamp that's easily replaceable with a set screw, and that's a common replacement part. The sample stream flows to the detector required for the chemistry. As the sample flows through, you'll see a peak rise on the FlowView software. After the samples pass through the peak, it'll settle back to a baseline until the next sample begins to flow through the flow cell. The peak is compared to the calibration curve, and a result is calculated against the curve. So here's a little bit of a breakout of the photometric detector with expanded range detection. Again, the lamp is up top. We have at the bottom right-hand side is where the nanometer filter is housed. Flow cell, we do have 5, 10, 15, and 20mm flow cells available. Say like for the low-level orthophosphate, you would be using the 20mm flow cell. The FS3700 detector design allows for the flow cell windows to be removed, cleaned, and or replaced if required without costly replacement of the entire flow cell. With the photometric detector, we've had refinements in detector design, improved signal noise ratio, and increased sensitivity. This led to minimizing noise, and we found that with an 8-port valve plumbed to utilize a bypass loop equal to size to the injection loop while in load position, it alleviated pressure differential and reduced detector noise related to the valve actuation. We'll move on to the FlowView software. It's nicely laid out. We have configure analyzer, build method, build sample table, run samples, view results, and perform maintenance on the instrument. It's intuitive software. It's 32 to 64-bit. It does work with Windows Pro 7, 8, and 10 Pro. We recommend Windows 10 Pro now since the support of Windows 7 has ceased. It performs dynamic auto scaling, keeping analyte peats on scale over a large dynamic range, 3 to 4 orders of magnitude. It allows viewing and editing results in real time. You can also add priority samples if you've got some emergency samples that come in. There's easy limbs, import, export configuration, reprioritize or edit the sample table on the fly during analysis, and mentioned earlier, software control of the cartridge heaters in 1°C increments. It's very easy to configure the analyzer. We can set up the pump. That's where you can set the run speed and timing of the peristaltic pump. Valves you can enable, disable for each channel and set timing for the valve load and inject. You can choose your channel, enable and disable channels, cycle duration, channel name. If it has a UV with total cyanide, you can enable or disable the UV digestion lamps if equipped. Heater enable, disable each and define heater set points if equipped. Detectors, you can configure the mode for photometric or amperometric. And then also for the sampler, you can set that up with a 180 position or 360 position. You can also set the depth of the needle going into the vial as well. Here we have the build method. That screen is divided into 4 sections that allow the user to define the method and calibration parameters assigned for each channel. Calibration table, the calibration, your signals, peak marking, and quality control. Next, what you would do is you'd build your sample table. This screen allows for you to define a sequence of samples to be analyzed. Then there is the open the run and start running your sample screen. Control the sample sequence and monitor data as it is collected. And again, this is in real time. You can then have view results. You open the review results screen for reviewing results and the data. The results may be reviewed from active or completed sequence. And here's a little bit more of a blowout with the view results. So here's your calibration results. We've got configuration tab, the methods tab, the calibration results, sample tab. Then also you can look at channel 1 and channel 2. We also have the maintenance screen. And this screen allows the user to view information and test the operation of all components attached, the FS3700, from a single screen. It may be accessed at any time and provides real-time information on raw detector outputs, FIA valve operation, UV lamps, heaters, and other devices. So this is an excellent tool used with technical support and looking for any potential problems in cycling the valve through operating the different heating zones, diagnostics, debugging, can work with the Autosampler as well. Some additional features of FlowView is a sample table import from CSV file. CSV file containing sample IDs, cup numbers, sample types, and other information may be imported into the sample build table screen. The user provided CSV file must conform to format compatible with the FlowView build sample table editor, and its customizable import format via the import screen. It's similar to the Microsoft Excel text file import. Have a full screen GUI, again, able to pause and resume the run between samples, add emergency samples, and we also support multi-languages as well with the FlowView. So now we're going to step into the chemistries that we supply and provide. Last week there was questions about the MDL operating range and such. So we have this document that covers the chemistry analyzer methods. This is available on our webpage. It's our document number 4075. So it lists out each analyte, technique that we use with this particular analyte, methods that it adheres to, the operating range, MDL, throughput with the samples per hour, our part numbers and such for channel, and then also for the cartridge. So again, we cover the ammonias, your chloride, your cyanides, all the different cyanides here that we cover. We've got hexavalent chromium, MBAS, nitrate, nitrite, nitrite milk, your phenols with inline and post-distillation, phosphorus all forms, phosphorus low level, sulfate, and then TKN. And again, as I mentioned, this is on our web page, www.oico.com, and you can look for publication 4075. So here is an example of what we do with these chemistries. We do have method abstracts that are also available on our webpage, and these gives you sort of a synopsis of the chemistry methodology. We again show range, a rate per samples per hour, precision, and then method detection limit, the MDL. So wanted to show example here of gas diffusion. We kind of go through by saying this is used for determining TKN nitrogen in drinking water, surface water, municipal industrial wastewater, according to EPA method 351.2. A gas diffusion step is used to separate the TKN from the particulates, ions, and copper-containing digest matrices. This method could also be used to determine ammonia nitrogen according to the EPA method 350.1. So again, we'd give a little bit of a synopsis by saying the sample pH is raised to a pH of greater than 11, and the ammonia molecules generated pass through the gas diffusion membrane and are absorbed into the alkaline hyperchlorite solution to form chloramine. The chloramine reacts with salicylate to form endophenol blue in an amount that is proportional to the ammonia concentration. Sodium nitroferrocyanide intensifies the blue color, and we measure that at an absorbance of 660 nanometers. So here's a little breakout of the chemistry as well. This shows the reagents and such that are used, and then shows that we do use the gas diffusion module. It is a heated chemistry, as we show 50 degrees C, and then measured with 660 nanometers. And we do have this available for all the chemistry on these method abstracts. And then a really neat little thing that I kind of learned is that you can see that by using the hyperchlorate, the sample, the complexing reagent, the salicylate, and the N2, that's a 5 pump tube chemistry. So you know that you'll be taking up 5 spots on your pump. Again, with these abstracts, we provide results as well for your review. Talk about digestion of DKN samples prior to analysis in the presence of sulfuric acid, potassium sulfate, and a copper catalyst at a final temperature of 380 degrees C. Free ammonia and organic nitrogen compounds convert to ammonium sulfate under these conditions. The digestion step is not carried out when you're analyzing the ammonia singly by the EPA method 350.1. And then we show results and a calibration curve with each of these abstracts. We have ammonia nitrogen phenate by US EPA 350.1. Again, giving you the method performance. We follow it by also standard methods 450 NH3H. And then also, again, we have that little breakout of the chemistry. So you can see that this is a 6 pump tube chemistry to take up 6 positions. It's also heated at 50 degrees C and measured with a 640 nanometer filter. We have chloride by the standard methods of 450 CIE. Here's cyanide post-distillation by EPA method 335.4. And this is the method where you would still use the distillation step first before running it on the instrument. This is used with a photometric detector. So in this application, this is a 4 pump tube chemistry heated to 337 degrees C and measured with a 570 nanometer filter. We have the nitrate nitrite and we developed that with FIA and segmented flow. Some states here in the United States require flow injection, and some say you have to run it with segmented flow. If remember from the prior presentation last week, a lot of the original methods were developed as segmented flow, and then several years passed, flow injection was then brought on. So we have this in segmented flow and flow injection following EPA method 353.2. We go through the scope and application, kind of break out what we do here. There's the nitrate plus nitrate nitrogen. We're using the cadmium coil. And then also, as we mentioned last week, between the differences of segmented flow, flow injection, with the cadmium coil, this is one of the methods that we have the injected flow with this. We need that because you want to have that dispersion going on within that reactor. If you're just doing nitrate nitrogen, you bypass the cadmium coil and run it as that way. Then we have the method with flow injection using the FIA valve, using your cadmium coil again, the bubbler, and 450 nanometer filter. And showing a little bit of the results. And what we also like to do on the methods is include at the bottom part numbers with consumables for replacements that are most commonly used with this instrument. So we provide part numbers and such for you for easy order placement. Again, breaking out results and showing calibration and graph of results. We have the phenol inline distillation following EPA method 420.4. With inline distillation, we also provide the distillation module with this apparatus and setup. So it can be done also by ISO method 14402. We have phenol post-distillation, where that is a distillation step is performed and then run through the analyzer. Here's phosphate all forms. So this is the chemistry that has the orthophosphate and the total phosphorus within the method. We give you how to set it up to run OPHOS, or then how to run it in total phos. So it covers all the phosphorus methods, follows EPA 365.1, also follows the standard method 450-PG, and also ISO 15681.1. Then we also have the low level that it follows with EPA 365.1. This also has the 20 millimeter flow cell, and this is getting down to the real low levels. I used this as an example last week as a municipality out on the West Coast uses this because they have to do 100% recycling of their water. So they have to monitor their phosphate at very, very low levels. So here with our method is an example of the valve, detector, and pump tube with rates for the method. We have here the 8-port valve configuration layout. This is total cyanide, by the way, so we have our flow cell, reference electrode listed out, common consumables for those. And then here is the chemistry plate that you see. So with this chemistry, this is 6 pump tubes. We use green, green, orange, orange, white, white, orange, 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 blue, and black, black. We list out all the different part numbers too for your mixing coils and such. And so there's your gas diffusion module, and then off to waste. So also too, within the methods, we have the recipe. So it tells you what pump speed you need to set it at. That's going to be at 40%. Your sample loop is a 200 microliter sample loop. Your bypass loop is a 200 microliter sample loop as well. And then also list out the tubing for each little, between each mixing coil. And then we also have the part numbers for your UV lamp that we use. And at this method for total cyanide, it's 312 nanometer lamp. Again, breaking out chemical consumables per chemistry. This is the total cyanide, and we provide that with the method for you to have the most common consumables that you need to purchase. Okay, so we have ISO methods as well. And these methods are for used in the determination of cyanide in groundwater, drinking water, surface water, leachate, and wastewater. We have the free cyanide by the gas diffusion and photometric detection following ISO 14403. And then we also have the ISO method for inline ultraviolet digestion, gas diffusion, photometric detection with ISO 14403. And this is quite a complex chemistry. As you can see with the diagram, a host of pump tubes, a lot going on. You're doing a distillation step. So prior to the analysis, you treat the sample, remove potential interferences. You use UV digestion, releases the cyanide from cyanide complexes. The acid addition converts cyanide ion to hydrogen cyanide gas, which passes under gas diffusion membrane. Then the hydrogen cyanide gas is diffused through the membrane and absorbed in a sodium hydroxide solution. The sodium cyanide is converted to a cyanogen chloride by reaction with chloramine T at a pH less than 8. And the cyanogen chloride then reacts with either these different compounds and such. So in this method, we have a sample prep guide showing you the different forms. Most commonly is the second one, the pyridine bariatric acid to form the red colored complex. The sorbate is measured at 570 nanometers. We have MBAS, the methylene blue active substances, using ISO 16265. This is used for determination of methylene blue active substances indexed in drinking water, groundwater, surface water, domestic and industrial. Ionic surfactants are the most important substances showing methylene blue activity. This method is therefore useful for estimating the ionic surfactant content in soaps. We came up with a really unique method for determination of nitrate nitrites in milk and milk products. Though nitrite and nitrate occur naturally in plants and soil and are essential for good health, too much can cause that baby blue syndrome we spoke of last week and is linked to birth defects. It's possible for nitrate and nitrite to be introduced to milk and milk products, including whey, powdered milk, and infant formula in the manufacturing process. Thus, dairy products should always be tested for these contaminants for QA/QC and for morting values for exported products. And so this method follows ISO 14673-3. For milk products, we use the cadmium reduction and flow injection with inline dialysis. It's also acceptable to cheeses, hard, semi-soft, and processed milk powder, whey powder, liquid milk, and milk-based infant food. And we have a document for this specifically 4389. Mentioned last week too, we do some tobacco methods that we developed. So we have potassium by flame emission spectrometry and continuous flow analysis. This method describes configuration, calibration, operation of the flow solution system equipped with a flame photometer, which is used for analysis of potassium in drinking water, surface water, saline, and domestic and industrial waste. We also have a volatile base in tobacco by online distillation and segmented flow, and that's for the volatile base in tobacco leaf samples. Total sugar in tobacco by segmented flow, used for determination of total sugars in the tobacco extracts. The reducing sugars for determination of reducing sugars in the extracts. Total alkaloids, and this is used for determination of the total alkaloids as nicotine in tobacco extracts. And also we look for chloride in tobacco extracts with the using a segmented flow for this method too. All right, we'll cover a little bit with the cyanide, ammonia, TKM by gas diffusion. Cyanide compounds are used in a wide range of industrial applications. Hydrogen cyanide is used for reduction of nylon and methyl mercaptolate plastics. Cyanide salts are used in metal plating baths for electroplating of brass, bronze, cadmium, copper, gold, silver, and zinc. There's other industrial processes employing cyanide, including petroleum refinery, steel production, microelectronics manufacturing, specialty chemical, and pharmaceutical production. Potassium or sodium cyanide are used in mining operations to leach gold and other metals from ore. Our first instrument was the CN3202, which we launched in the mid-90s. And that also then followed the OI 1677 available cyanide method that the EPA approved in 2000. So from our models 3202 to CN3000, CN3100 to the FS3700, Alpchem, OI Analytical are the innovators in cyanide analysis. A really good guide for the cyanide methods is the ASTM D2036-09, and it's the standard test methods for cyanides in water. An excellent reference for wanting to learn about all the different applications and methods for cyanide. I kind of like to call this the Bible of cyanide info. Then there's ASTM D7365. It's a standard practice for sampling, preservation, and mitigating interferences of water samples for analysis of cyanide. And this talks a lot about, again, the sampling, preservation, and mitigating the interferences of water. This is where the D6888 method talks about adding the acid to help abate sulfide up to 50 milligrams per liter. You can use a lead acetate strip. If you continue to have more than 50 milligrams per liter, you need to dilute the sample. So with cyanide methods, we measure the various cyanide species. You go from free cyanide, that's the weak to moderately strong metal complexes, to available cyanide, which are the strong in between, and then you get to total cyanide, which are the strong metal complexes, and that's where that will require the UV digestion to help break those bonds apart. So free cyanide refers to the sum of HTM and CN ion in a sample. Free cyanide is bioavailable and about 1,000 times more toxic to aquatic organisms than it is to humans. Weak to moderately strong metal cyanide complexes are compounds that dissociate and release HCN under mildly acidic conditions. This is the weak acid dissociable, CATC, and OI method 1677, along with D6888. These methods are developed to quantify available cyanide, and these measure the weak and moderately strong metal cyanide complexes plus free cyanide. Strong metal cyanide complexes require strongly acidic conditions to dissociate and release hydrogen cyanide gas. Strong metal cyanide species include complexes of iron, cobalt, and platinum metal groups. The EPA method defined total cyanide D7511 includes the strong metal cyanides, weak to moderately strong metal cyanides, and free cyanide. The EPA defined total cyanide does not include thiocyanate and organic cyanides. So this is just a breakdown of our EPA method OA 1677, available cyanide. It's a method for determination of available cyanide in water and wastewater by flow injection, ligand exchange, and amperometric detection. We have the D6888. Sulfide is a known interference. and can be eliminated by use of complexing agents described in this method prior to the gas diffusion process that prevent hydrogen cyanide from diffusing. That reagent is a Ta2SAR. We have, I briefly touched on last week, the sour water application. That is with cyanide generation and corrosion, cracking organic nitrogen compounds and petroleum feedstocks, liberates hydrogen cyanide, ammonia, and other nitrogen compounds. The formulation and downstream effects of cyanide are a major concern in fluid catalytic cracking and hydrocracking operations. So we developed this application note, and this follows like with the ASTM method D6888. It's our publication 3741 and have that available on our website. Here's the total cyanide. So with ASTM D7511 uses narrowband low watt UV radiation to decompose cyanide complexes in samples at ambient temperature in a continuously flowing acidic stream. Reducing and complexing reagents combined with an ambient temperature UV digestion minimizes the formulation of matrix interference. Elimination of the sample distillation step enables measurement of cyanide at lower concentrations with improved precision. We also have a white paper on that. It's our white paper number 3687 and covers total cyanide analysis of NPDES wastewater samples using ASTM method D7511. We also have a application on soil and sediment samples. Insoluble cyanide complexes such as Prussian blue are bound to particulate matter or soil particles and are not quantitatively recovered by distillation procedures. A sodium hydroxide extraction followed by total cyanide analysis using ASTM D7511-2 recovers the cyanides, and that's our publication number 3944. And finally, we can also do simultaneous analysis of available and total cyanide. That's where both of these are run together. That's our application note 3793. Just wanted to speak a little bit as well with our expertise in cyanide from the lab to an online instrument. We do have the Model 9310 analyzer, which can be used as a benchtop analyzer for grab samples or deployed for online measurements and process control. It measures available cyanide and precious metal leaching solutions by EPA method OI 1677 and ASTM D6888. We have measurement ranges of 0.2 to 50 ppm, 2.0 to 500 ppm, and 20 to 2000 ppm. Little breakout of the instrument itself. It utilizes the gas diffusion module, the Amperometric flow cell, and reference electrode as well. It supports measurement and control of cyanide in multiple cyanidation unit operations. There's cyanide addition, leaching, cyanide recycle, detoxification, and effluent and discharge tailings. With our gas diffusion ammonia, the method update rule passed in 2012 with the EPA modified the 40 CFR Part 136 and allows diffusion in place of distillation for the analysis of ammonia. Utilize the continuous flow automated diffusion that passes ammonia through the hydrophobic membrane into an absorber solution that is automatically color developed and measured by the continuous flow analyzer method. Gas diffusion eliminates the need for distillation. This allows facilities with NPDES permits to use gas diffusion to test wastewater samples for TKN, ammonia, and the Clean Water Act compliance reporting. Higher sample throughput, lower labor cost, lower cost per analysis for the reagents and consumables. We also have this as a white paper, and that's our white paper number 3994. Again, all these are available on our website. I wanted to show our gas diffusion module, very simply designed with simplicity. The purpose of the gas diffusion module is to function as the inline cleanup, eliminating particulates, potential ionic interferences, and chemical complexes. The target analyte permeates through the membrane into a separate flow path going on to further analysis while all the other components of the stream go off to waste. Without separation, these interferences can cause problems with both amperometric and photometric flow cells by disrupting the electronic potential of the target molecule, causing precipitation in the colorimetric reagents, or simply creating problems with large particulates. So this is a layout of ammonia TKN with our gas diffusion module. And then this is the OI Analytical gas diffusion module for our cyanides, OI 1677, D6888, and D7511-12. So with gas diffusion, you get to say farewell to distillation. So there's no more macro distillations, no more midi distillations, no more glass, no more hazardous waste, no more hazardous waste disposal fees and such. These are really great tools to help with the lab and productivity. Here in College Station, we have a staff based here for assisting you. I'm the Senior Sales Specialist. We also have Tim Smith, who's our ACA Product Specialist with technical support, and also Mark Terrell, our Product Manager here at OI Analytical, and we're all based here in College Station, Texas. So I'd like to thank you again for joining us today. For more information, you can contact me at hank.hahn@xyleminc.com or info@apac@xyleminc.com. So again, thanks everybody for attending part 2 of the FS3700 presentation.
Hank, thank you very much for your time. That was a fantastic presentation. And we've got a few minutes to spare here, so I'd love to open this up to the audience and allow them to ask you some questions. So again, for those of you who are new to GoToWebinar, there is a chat or question box on your GoToWebinar interface. Simply type in your questions there. And again, if we run out of time, Hank will follow up offline and answer your questions. So Hank, we've already got a number of questions, so let's jump into this. The first one is what is the minimum sample limit for the flow solution analyzer?
Oh, minimums for the injection into the instrument. It varies per the chemistry. I don't have that handy, but if could email that and ask for which specific chemistries and such, could respond to you that way. I apologize, I don't have that.
Not a problem. In our audience, Jenna, if you've got the time, please use the email address that's on the current slide and perhaps send an email directly to Hank and he will support you with that. Moving on to our next question. They would like to know the name of the software that's associated with the analyzer.
That's our FlowView software, and that's software that we have developed specific with the FS3700. We did have a prior software with our other instruments called WinFlow, and so basically we kind of took the best of WinFlow and incorporated that into FlowView. So what's nice about FlowView software, again, it's a 32 or 64-bit, runs on Windows 10 Pro, and so that's the software we use with the instrument, and that ships standard with our FS3700. It's not like an add-on piece that you have to have quoted for you.
And on that note, Hank, is it possible to save the data as a project?
Oh, yes. You can save the data on a laptop. It can be exported to LIMS. So yes, all data is saved and stored.
Is the software compatible with Mac?
No, unfortunately, it's not. All of our software is developed on the Windows Microsoft technology.
Great. Another question here concerning applications. Well, what are some of the uncommon or rare application cases that you've heard about from various customers?
There has been seawater methods and such, looking at nutrient analysis. We have had in the past nitrogen, ammonia, phosphorus, urea. As I mentioned, OI kind of started with our TOC analyzer with Texas A&M University and their oceanography program. There's some interest in oceanography chemistries, also soil plant, and had nitrate, nitrite, ammonia, and your phosphate with those chemistries. Rare occurrences and requests, and we are developing methods for sulfate, also sulfide as well.
Terrific. Thank you, Hank. Well, that's all the questions we have for today. So before I give you the floor for your final comment, I'd just like to remind everyone that there will be a follow-up email coming out of GoToWebinar that will include a link to this recording as well as a link to a certificate. And again, we have many more webinars available on our Stay Home, Stay Safe series, which is accessible via our website, which is currently on the slide that's before you. Hank, thank you again. I leave the last few words to you.
Thank you again, Michael, for the opportunity to do this series on the FS3700, and really enjoyed being able to speak with emerging markets and folks across the world. So thank you so much.
Thank you, Hank. Thank you, everyone. Be safe and have a great day. Bye-bye.